The overall goal of this procedure is to record the synaptic and excitability properties of embryonic zebrafish Mauthner cells. This is accomplished by first dissecting the zebrafish embryo to expose the ventral surface of the hindbrain. The second step is to place the dissected preparation onto the stage of a patch clamp microscope and identify the Mauthner cells. Next, patch clamp onto the M cells using the appropriate recording mode and record synaptic activity and excitability properties. The final step is to analyze the recorded data to ascertain synaptic or excitability properties. Ultimately, patch clamping from these cells is used to show the properties of synaptic receptors in a developing organism.
Generally, individuals new to this method will have two main areas of difficulty. The first is the dissection. The second, formation of the gigaohm scene, can be challenging. I first had the idea for this method when I decided to work on a zebrafish preparation in which the hindbrain was attached to the spinal cord. Demonstrating the procedure will be Birbickram Roy, a PhD graduate student in my lab.
Begin this procedure by preparing the dissecting dish lined with Silgard. First, fix the washers to the slide with grease. Then, add liquid Silgard to the center of the washer. The Silgard usually cures overnight and forms a small circular bed on the glass slide, which becomes the base of the dissecting dish. After placing a cover slip on the slide, place the slide into the recording chamber to serve as the base for the dissection. Afterward, prepare the required solutions. The extracellular solutions should be left at room temperature, while intracellular solutions should be kept sterile. One can also add 0.1% lucifer yellow to the intracellular solution.